on my end. So we're getting underway here on Tuesday the 24th of September. We are going to be moving into part 2 of our course. So we'll be moving into the section on sensation and perception. That's today and Thursday. Next Tuesday we will be covering in a one-day, one-session sort of way, the section on consciousness. And then we have 2 sessions each on learning and memory, with test number 2 rolling around on Thursday the 17th of October. There was something a little funky going on with Collaborate in my first session, and it seems like that is going to persist in this section. So my workaround was to just do an app share where I show you my PowerPoint slides as they reside on my desktop. I could not get the PowerPoints to load into Collaborate this morning for some reason. Not sure why that was, but that's just the way it goes. Sometimes this technology doesn't always cooperate, so we'll work around it by doing basically a session-long app share for you guys. So let me start that on my end. Press the buttons to make the magic happen there, and I'm going to toss my desktop out to you guys. And good. I want to jump over here into PowerPoint where I've got my list of difficult items for test number 1. So we're just sort of, you know, using duct tape here to make happen what we want to have happen, which is me showing you the slides here. So this is the first of the 6 items from test 1 that caused the most problems. I'm just going to review these with you guys and point out some things that hopefully will help you going forward. Number 1 here says, plasticity refers to the brain's capacity to do what? Well, the correct answer here is C, build new neural pathways. Okay, so where did this one come from? This was a textbook item. That is, it's an item that was not covered during lecture, but was covered at the end of Chapter 2, the bio chapter, and plasticity is one of the ideas that we're going to want to have with us as we go forward, studying the different biological underpinnings of different aspects of behavior, mental processes that will be important going forward. Question number 2 was this one. This was actually the most difficult item on the test because only 25% of you got this one right. So let's take a look at this. If psychologists discovered that people who live at the poverty level have more aggressive children than wealthy people, what would this indicate? Well, A says poverty has a negative influence on children's behavior. That sounds to me like they're talking about causality, right? So let me type in here causality. Right? They're talking about poverty causing changes in behavior. Well, that's not right. The factors that lead to poverty also cause aggressive behavior. Well, that actually says the word cause in it, and we know that correlation doesn't mean causation. So it's not A, it's not B, so therefore it can't be D. So we're left with C. Let's look at C. People's economic status and aggressiveness of their children are negatively correlated. That is, as economic status goes down, thumbs down on my left hand, what happens with aggressiveness? Aggressiveness goes up. That's exactly what it says up above. People who live at the poverty level have more aggressive children than wealthy people do. There you've got this scenario. Choice C is the correct answer there. Okay, any questions on that one at all? And I'll pop open the chat over here just so I can see hands or other things going on. And I'm actually following along on my iPad to make sure this doesn't do anything crazy as we're trying to run the session in this odd way today. We'll get the problem figured out for next week. All right, third question here. The chemical messengers of the endocrine system are called what? Well, the endocrine system has to do with hormones. So that was a textbook question, but we actually talked about the endocrine system when we discussed the hypothalamus, right? We said the hypothalamus was in charge of the pituitary gland, which is the master gland of the endocrine system. The endocrine system runs via these hormones. So that item was answered correctly by about half of the students in the class. Next up was this one on the difficult item list, a clinical psychologist who explains behavior in terms of unconscious drives and conflicts. That's all I need right there. Unconscious drives and conflicts. As soon as I see that, that's got to be Freud. That's got to be psychodynamic as the correct answer for that one. Okay? Number 5, a correlation. Again here, another correlation question. About half of you got this right. Between levels of impulsiveness and annual income of negative 0.75. So we have to be able to interpret a negative correlation. What would that mean? Well, if you look at A and B, what are A and B? Tell me in the chat box. This is kind of like the split brain patient question. A and B are what? They're opposite. So what do we think here? Probably one of those is right. So let's take a look here. A says lower levels, thumbs down, of impulsiveness are associated with lower levels of annual income. Both thumbs are down. They're moving in the same direction. That's not a negative correlation. Because a negative correlation is when things move in the opposite direction. Let's look at B. Higher levels of income are associated with lower levels of impulsiveness. That's exactly right. B is the way to go with this one, right? We've got a scenario where higher levels of annual income are associated with lower levels of impulsiveness. Okay. Questions on that one at all? All righty. And our 6th and final question here to review together was this one about Parkinson's. The tremors of Parkinson's disease result from which neurotransmitter deficits? Well, that has to do with dopamine, and about half of you were able to get that one right. We talked about dopamine both with respect to schizophrenia as well as Parkinson's. Parkinson's is what Michael J. Fox has. The tremors result from decreased levels of dopamine in the substantia nigra. Okay? All right, let me pop up the chat for a second. Questions or comments about any of those items or about Test 1 in general? Anything in Test 1 that you loved or hated, you thought was fair or unfair? Not a whole lot of anything. All right, let me grab the slides that I've got for you here on sensation and perception, because we're going to move into that as the first segment of part 2 of our course here. So we're talking about sensation and perception this morning. And I'm going to start off this discussion by helping you distinguish between sensation and perception, so that this contrast between these two different parts of how we process sensory information makes sense. So sensation, I'm going to say, is both earlier and more biological in its focus. Okay? Earlier and more biological is what sensation is going to be about. And to contrast that with perception, we can say, well, perception is later, not surprisingly, and rather than more biological, it's going to be more psychological. Okay? So perception is going to have to do with psychological processes, whereas sensation is going to be more focused on biological processes. The easiest contrast there that I can help you use to understand this distinction is the fact that today, when we're talking about sensation, which is earlier and more biological, we're going to be talking about parts of the brain, parts of the visual system, parts of your auditory system. Okay? Looking at structures, looking at biology. Thursday, what you'll see as we discuss perception, there's not going to be any discussion of brains or receptor cells or any kind of physical biological hardware. Okay? It's going to be about, when we talk about perception, how your experience of new stimuli is influenced by previous experiences you've had being exposed to other types of stimuli. Right? How we interpret and understand new information coming in from our environment, that's largely a function of what we've been exposed to previously. Okay? So we'll be using that perception framework on Thursday. Today, the focus is on sensation. And I'm looking at the list of learning outcomes here from the syllabus, and it says, number 1, for sensation and perception, Chapter 6, describe the operation of sensory systems. That's today. That's sensation. That's like parts of the eye, parts of the ear, as we review the visual and auditory systems respectively. Okay? This will make more sense once we work our way through today's discussion And then when we work our way through Thursday's class, you'll see, I think in even more stark contrast here, the distinction to be made between sensation and perception. Okay? All right, let's take a look at the next slide, which has a little question for you. So I want you guys to take a shot at this one. And the way you respond to this one is on the left side here. Once I make it so, you'll be able to roll over where it says check mark or X right now. You'll be able to respond A, B, C, or D. So let me make this into an A, B, C, D multiple choice. We'll get this started. So take a look at this question. You find that you have to add 1 teaspoon of sugar to a cup of coffee that already has 5 teaspoons of sugar in it to notice the difference in sweetness. If you have a cup of coffee with 10 teaspoons of sugar in it, how many teaspoons would you have to add to notice the difference in sweetness at least half the time? This is one of these pre-teaching questions, right? I haven't taught you anything yet. I'm going to ask you what you know about it now. I'm going to teach you some things, and I'm going to ask you the same question in just a bit. And at that point, hopefully what will happen is a lot more of you will get it right than are getting it right this first time. So I'll give you guys another 20 seconds or so to take a shot at this one, and then we'll take a look at your responses. Okay. You can see over here the responses thus far. 29 of you think it's going to be, 30 of you think it's going to be B. 8 of you think it's going to be A. 6 of you think it's going to be D. So let's table this question for right now, and let's come back to it in just a bit. Okay, we'll come back to it after we've had a chance to talk about some of the basics here about sensation. So sensation, as we get started here, is actually part of the little subdiscipline within psychology that's referred to as psychophysics. So if I study sensation and perception, I could technically say I'm a psychophysicist. So what is psychophysics about? As it says on the side here, it's the study of the relationships between the physical characteristics of stimuli, sights and sounds and smells, including things like the intensity of those sights and sounds and smells. The connection there, the relationship there between that stuff and our psychological experience of those things. Okay? So we're exposed to various sights and sounds and smells on an everyday basis, and psychophysicists try to study how those stimuli influence ourselves, how they influence us psychologically, maybe make us feel happy or make us feel sad or afraid. How we process these things, again, both at a level of sensation on the front end, earlier and more biological, and perception on the back end, later and more psychological. And we will wrap up this morning by looking at where we're going to go on Thursday as we talk about perception. Okay, so psychophysics is the term that's used for this little subfield within psychology. Two key terms on this slide are absolute threshold and just noticeable difference. I guarantee you I'm going to ask you a question about at least one of them. So let's just determine what each of these things are about. Absolute threshold, it says here, is the minimum stimulation needed to detect a particular stimulus at least 50% of the time. That means getting it right at least half the time in terms of saying whether something was present or not. What I want you to understand an absolute threshold as is it's a change from 0 or a change from nothing. Okay? So here's what an absolute threshold is. It's you sitting in a completely darkened room, and someone has a light switch control in the adjoining room that's a dimmer switch, and lets them turn the light a little bit brighter, a little bit brighter, a little bit brighter. The first point at which you notice that the room is no longer completely dark would represent your absolute threshold for vision. Okay, so as it says on the slide now that I typed in, it's a change from 0, it's a change from nothing. There was nothing present in terms of visual stimulation, and now there is something present, right? If you think back to grade school when you had your hearing test, right, and they played different sounds in your ear. The first time you heard that really low-pitched sound, let's say, Okay, maybe at first you didn't hear it, and then they increase the volume just enough for you to be able to notice it, and you raise your hand. The first time you raise your hand would represent your absolute threshold for hearing. In fact, I can give you the real-world example that relates to hearing, and it is this. The absolute threshold for hearing for human beings, on average, is the ability to detect the ticking of a watch, obviously an analog watch with hands on it, right? A ticking watch at 20 feet from you in a completely quiet room. Okay? You can get that right. You can notice that. You can detect that at least half the time. Ticking watch 20 feet away in a completely soundproof room. Okay? If you want another sense, and another absolute threshold example, we know that the absolute threshold for taste is the presence of 1 teaspoon of sugar in a 2-gallon jug of water. What does that mean? Well, that's the first point at which you notice that that water with the 1 teaspoon of sugar in it is sweeter than the water that comes from this other 2-gallon jug of water, which has no sugar in it. Right? So we set up 2 2-gallon jugs. One is just water. One is water plus 1 teaspoon of sugar. The second one tastes sweeter. You notice that at least half the time. There's your absolute threshold. Right? It's a change from 0 or nothing being there. Right? In that case, in terms of an added sweetness to that drink. Okay? That's an absolute threshold. What's a JND? I love JNDs because the term basically defines itself. It's a just noticeable difference. So what does that mean? It's the minimum difference between 2 stimuli that's required for detection at least 50% of the time. It's sometimes referred to as a difference threshold. I like to think about a JND as a change from one thing to more or less of that thing. Okay? A change from one thing to more or less of that thing. Does that make sense? It should, right? So something was present, and now there's either more of it or less of it, and I notice that change, a just noticeable difference. The thing could be sound. Let's run with a sound example. Okay? Let's say that you're at a party and the volume on the stereo reads 20. Okay? And you want to see what your friend's JND is for the loudness of the music being played from the speakers connected to that stereo. Okay, great. I turn it up to 21. She doesn't say it's any louder. 22, no. 23, no. 24, she says it got louder. Okay? Tell me in the chat box, when you go from 20 to 24 on the stereo speakers, what percentage increase is that from 20 to 24? I need the math majors to chime in. When we jump from 20 to 24, the increase there is what? Don't all jump at once. It's a 20% increase, right? It's 4 over 20, which is 1 over 5, which is 20%. So Alexandra and Marissa get 8 million bonus points. So what we're saying there is it's a 20% increase there in that example that represents your JND. That's just a made-up example. It's not necessarily 20%. I'm just trying to give you one to think about, right? So if we use this idea to understand Weber's law, let's think this through. Weber's law says JNDs are proportional to the size of the original stimulus. JNDs are proportional to the size of the original stimulus. So let's run with this 20% increase example for hearing. And let's say that instead of starting at 20, let's say the original stimulus for the sound was 30. To what level would I have to turn the volume for you to notice a change from 30? It would have to go from 30 up to what for you to notice that it got louder if we apply that 20% increase rule? It's got to go from 30 up to, what's 20% more than 30? It's not 34. 36, right? So we have to go 1/5 higher. Well, 1/5 of 30 is 6. 30 plus 6 gets me to 36, right? JNDs are proportional to the size of the original stimulus. Let me do one more example, and then I'll go to Courtney for her question. Okay. Another example, and it's not really sensation. I guess you could argue that it's touch, but just to give you another scenario, let's say you're holding a box of books that weighs 10 pounds. And I put a little more weight on that pile of 10 pounds of books. And you first notice the increase, let's say at 13 pounds. Okay. What percentage increase is it when you go from 10 pounds to 13 pounds? How much heavier is that? It's 3 pounds over the 10 that we started with, so that's a 30% increase. So if we apply Weber's law, what kind of increase will we be talking about if you were starting with 50 pounds of books in your hands? How heavy would it have to be for you to notice that increase in weight? It would have to go from 50 to what? 65. Really good. I like the math that you're doing too, right? That's 15 over 50. That's a 30% increase. We increase from 50 to 65. We notice that it has gotten heavier. Okay, that's the idea here of Weber's law, right? J and D's are proportional to the original stimulus. Let's go to Courtney for her question. That can go right into the chat box, and I'll just follow it on the iPad here. Can you explain the 20% increase? Why is it 20% for only 4 notches? Well, it was the example that I used, right? So I said we went from 20 to 24. That's a 20% increase because 4 over 20 is 1 over 5, and 1/5 is 20%. I just made up that example. It could be an 8% increase. It could be a 17% increase that you notice. Whatever the specific scenario is, that same percentage increase would apply to all subsequent examples, right? So if it was a 20% increase for the volume, if the radio was cranked up to 60, we need a 20% increase from 60, which I think is 12 more, so we'd have to go to 72. That make sense now? Good. All right, I see that you said you've got it. Fantastic. All right. I want to tie these two concepts together for you by saying that really, if you think about it, an absolute threshold is really just a JND from 0 or nothing. Does everybody see that? That idea makes sense, right? Just noticeable difference from 0. That's what an absolute threshold is. If a JND is about a change from some amount of something to more or less of it, an absolute threshold is a JND from there was none of it, and now there's been a change to a level that I can detect it again at least 50% of the time. Okay? Good, good. Any other questions? Courtney, I got yours right, so if you click your hand, that will lower your hand so I know that you don't have additional questions. Thanks for that. Anybody else with questions on this first set of concepts of absolute thresholds, JNDs, and Weber's law? It's pretty straightforward, but it's important stuff for sure. Okay? All right. Let's go back to the sugar question. There it is. So take a second shot at that. I'll get a quick drink of water, and we'll see how you do the second time around here. Okay, I'm going to give you guys another 10 seconds to finish up here. Now look at us. 36 out of 48. Now 75% of you are going with B. B is correct. Why is it B? Tell me in the chat box in your own words here why the correct choice is B. 2 teaspoons. Right? It's a 20% example. It's not always going to be 20%. It just happened to be that. So we went from, I think, about 60% of you guys getting that right to more than 75% of you nailing that question. So good. I will leave that then where it is and move us on to a consideration of the different sensory systems that we're equipped with, right? Vision, hearing, smell, taste, and you're probably assuming the fifth one would be touch. Touch is actually going to get subsumed under the somato senses down here. Somato means body. So these are the body senses, including touch. Your vestibular sense, which has to do with balance and sort of dizziness, right? So if you've ever spun around, you stand and you put your forehead down on a baseball bat and spin around and around and around and around, and then you stop, you get dizzy and you fall down. It's because of the movement of fluid in your semicircular canals in your inner ear. That's your vestibular sense. Kinesthesis has to do with your positioning and your movement of your muscles and joints, so we're not really interested in that all that much. In fact, here's how we're going to approach this, guys. We are going to spend our time this morning talking about vision and hearing. So our last 50 minutes or so here, 45 minutes, is going to be a review, pretty thorough review of vision and hearing. Your job is to read about the remaining senses in the textbook. Okay, smell, also known as olfaction, taste, also known as gustation, as well as these somatosenses. The only thing I'd add here, just to get you started on smell and taste, is that both of these guys, smell and taste, are referred to as the chemical senses. Okay, the chemical senses, because they have receptors that are attuned to not forms of energy, as is the case with vision and hearing, but instead they're responding to chemical molecules. Okay, so vision, we're going to learn, responds to forms of electromagnetic energy. Hearing responds to sound waves. Okay, in contrast, smell and taste are your chemical senses. So your job is to read the textbook here on the remaining senses. It's a pretty good bet that if I throw a couple more textbook-only items onto test 2, one of them might be on one of these other senses that I'm not going to cover in lecture. And the reason I don't cover the other senses is not because I think they're unimportant, but once we cover the ground that we cover with vision and hearing, it's pretty thorough. It's more than enough information to understand those two, and you could sort of map that model, that framework, onto these other senses. And that model actually begins with our next slide, which has two of the really big ideas really for the whole class session today, and that is the concepts of receptor cells and transduction. Okay, so if you're not paying attention right now, this will be a good time to turn around and start paying attention, because these related ideas of receptor cells and transduction are very important as we go forward and talk about vision and hearing. So what's the deal here? Well, we have specific receptor cells for each sense that stimulate neurons in the CNS. The CNS, you should remember, is what? The central nervous system. Okay, so we've got receptor cells for vision. We've got receptor cells for hearing. We're going to talk about those this morning. What do? Well, it says down below here, they transduce sensory input from the environment. Sights and sounds and smells, they transduce. What does that mean? Well, transduction is a process of conversion or translation, and what they do is they translate or convert or transduce sights and sounds and smells. They turn those things into what? Action potentials. APs, action potentials. So transduction is the process whereby receptor cells for, let's say, vision convert light waves into action potentials. Okay, think about that for a second. We've got a system here. We've got specialized cells that turn light waves from the world around us into action potentials. Now, that's really cool. The question is, why does that matter? Why is that important that we have receptor cells? Well, the answer is, without receptor cells, you wouldn't be able to see anything for vision. Because here's the story, guys. If I were to open up the back of your skull, and I wouldn't do that without asking your permission, and I were to shine a light onto the primary visual cortex in your occipital lobe there in the back of your head. Tell me in the chat box, are you going to see anything if I just shine a light directly onto the visual cortex? Well, obviously the answer is no, because of the way I asked it. Why won't you see anything when I shine light onto your brain there in the back of your head? You should see something. It's your visual cortex. No, your skull is opened up. I'm shining it directly on your brain. You don't stimulate receptor cells. It takes in from the front, says Ashley, right? The reason why is those cells in the brain, in the back of your head, they speak the language of action potentials. That's the way to think about it, right? Action potential is the language of the central nervous system. Okay? So what we need in the front of our head, where the visual information comes in through our eyes, is some kind of a system to convert, or here the word is transduce, right, light waves into action potentials. We have them. We're going to talk about them in a bit. They're called rods and cones. Without rods and cones, you can't convert, you can't transduce light waves into action potentials. Okay? You need that translation to occur. Okay? Again, because it's turning the light waves into the language, if you will, of your central nervous system. And that language is action potentials, right? Electrical within, chemical between. We know all about that. Okay? So let's take a look at the sensory inputs for our visual system. And the sensory input comes in the form of electromagnetic radiation. And the point of this slide is to show you that our visual system is attuned to a very small sliver of the full electromagnetic spectrum, right? From about 400 nanometers over here to the left to 700 nanometers over here to the right. Here down the bottom we see the full electromagnetic spectrum laid out in wavelength in nanometers, billions of a meter of the light waves. Well, we see visual light between about 400 and 700 nanometers. At the shorter end of that wavelength spectrum, we have the purples and the dark blues. On the longer end of that electromagnetic spectrum that's visible to us, this visible spectrum, 700 nanometers is the reds and the oranges. Okay? We don't see X-rays. We don't see ultraviolet rays. You don't see the AM or FM broadcast bands. If you do, then that's kind of not good. Right? But you won't. You won't see those because your brain is not built in such a way as to be able to process that segment of the electromagnetic spectrum. So what I want to do now is walk you through the hardware. Remember, we said at the beginning today, this is going to be earlier and more biological. Thursday will be later and more psychological. Okay? So let's take a look at some of the biology that underpins our visual system, and that has to do with the major parts of the human eye. So we're going to review about 7 or 8 major parts of the eye on this slide and the next couple of slides. By the time we're done doing this, you should be able to go to your optometrist next time, and when the doctor takes a look at your eyes, be able to tell him, hey, that poster over there, I learned about all those parts of the eye in psych. All right, so light waves enter the eye through the cornea. So we're looking at an image of a candle. The first structure we want to talk about is the cornea right here. The cornea is the transparent covering over the eye, which serves partly to focus light on the back of the eye. So you can see this image ultimately getting projected onto the back of the eye right here. Okay? So the cornea is this transparent covering over your eye. You could scratch your cornea. So if you go to, itch something near your eye, you accidentally stick a fingernail into your eye there, you could scratch the cornea, this transparent covering. Pupil and iris go hand in hand. The pupil controls the amount of light that enters the eye. It's the opening in the iris. Okay? So the pupil size is controlled by the muscles in the colored iris. So let's figure out what happens with your pupil depending on whether it's bright and sunny like it is outside right now, or it's dark like it would be at night. Okay? What happens to your pupil when you go out on a bright sunny afternoon like we're having today here in San Diego? Or I guess I should say a bright sunny morning. Your pupil does what? It certainly gets smaller. It constricts, as Aaron says. That's the fancy term for that. As you then go out late at night, you're out in the desert looking at the stars, obviously it's going to do the opposite, right? It's going to get bigger. It's called dilates, right? Your pupil will dilate in a low light environment. Why does it dilate? It opens up. It opens up to let in as much ambient light as is available to help you be able to see as well as you can. Okay, so the pupil size is controlled by the muscles in this colored iris. So if you have brown eyes Or if you have blue eyes, that colored muscle there that you're saying, I have brown eyes or blue eyes, is the iris. Okay? So cornea, transparent covering, pupil and iris hand in hand. Pupil dilates to let in more light when it's dark. It constricts to let in less light when it's really bright. Here's the lens. You can see it here. It's shaped like a lens. What's that? The lens is an elastic structure that sits behind the pupil, and the lens will become thinner to focus on objects in the distance. So I'm looking out my back window right now, and I can see some houses down a block or two. My lens, as I'm looking at those distant objects, is becoming thinner. And then as I turn around and look at my notes in front of me here on my kitchen table, it will become thicker to focus on those nearby objects. Okay, that's what the lens does. Thinner for things far away, thicker for nearby things. The lens becomes less flexible. It becomes thicker and less flexible as you get older. So with age, the lens will thicken and become less flexible. And that often causes older folks to have trouble seeing things nearby. So if you've noticed over time that your parents, when they're reading something, are extending their arm farther and farther, okay, away from their eyes, to help them be able to see, that has to do with the decreased flexibility in the lens here over time. Okay? Any questions on that much so far? The front side here, cornea, iris, pupil, and lens. What does the lens do? Well, focus the image then on the retina. So this retina is going to be really, really important. I put a star next to it in my notes if I were you. Okay? The retina is a sheet of tissue at the back of the eye, which is about as thick as a piece of paper. So you see here this sort of tan-colored structure. It's almost shaped like a backwards letter C on the slide here, lining the back of the eyeball, the retina. Okay, the retina is so important, and you should stick a star next to it because what does the retina contain? The retina contains the receptor cells for vision. Okay, they're sometimes referred to as photoreceptors to denote the fact that they're responding to light waves. Okay, so the retina contains the receptor cells for vision. That's why it's so important. Well, what's the significance of that? Because those receptor cells are what are going to convert light waves into action potentials. Okay, they turn the incoming stimulation from your environment, again, into the language of your central nervous system. They turn light waves into action potentials. They're called rods and cones. Let's go to this next slide, and we'll come back up. Okay, rods and cones are the two kinds of receptor cells for vision, and they work in complementary ways. Look at the rods. The rods are more concentrated in the periphery of the retina. The periphery is just a fancy way of saying the edges of the retina. So we had that sort of backwards shaped C on that last slide. At the top of the C and at the bottom of the C, we'd have a greater preponderance of rods than cones. Okay, rods do black and white vision, and they're more active in dim illumination. Okay, so when you're out at night in the dark backcountry here in San Diego County looking up at the stars, you'd have rods being more active. If you were sitting in a movie theater and the movie was just about to start, they just dim the lights, your rods would become more active in that situation. Cones do complementary things here. So what do cones do? Cones are concentrated in the center of the retina, and they do color vision. So we see cones more in the center. In fact, there's an area that we'll look at on the previous slide in just a second called the fovea. The fovea is a unique spot on the retina because the fovea contains cones and only cones. There are no rods at all, no rods whatsoever in the fovea. Okay, so we've got cones concentrated in the center of the retina. Cones are responsible for color vision. Okay, and you'll read more about color vision in the textbook as well. But as we process colorful images or images in bright illumination, that's when we're going to see activation in the cones. So you can see via this slide how we sort of have this complementary set of roles played by the rods and the cones as we process visual information. Okay. Rods doing black and white and dim illumination, cones doing color vision and bright illumination. So I've got a mnemonic for you here for remembering cones at least. Cones begin with C. They're concentrated in the center of the retina, and they're responsible for color vision. 3 C's, right? Cones, center, color. You shouldn't forget that now, right? Cones, center, and color. So let's go back up to the previous slide for a sec. So we're talking about the specialized cells here, the receptor cells for vision. They're located in the retina. We've got more rods here and here at the top and the bottom. We've got more cones through the middle here. In fact, at the fovea here, let's especially call this out. The fovea is, again, the spot where we have cones and only cones. Okay? Any questions about the retina or rods or cones or the fovea? We're now on the back side of the eye, right? A couple more things to point out. We actually have a blind spot right here, right at this location where the optic nerve is exiting the back of the eye. Why do we have a blind spot there? We have a blind spot at this location, which is sometimes referred to as the optic disc, O-P-T-I-C, D-I-S-K, optic disc. Try to send that out to you guys here, optic disc. Okay, when it says clicker 75, student number 2, that's me on the iPad. So that I don't have to bang around on the computer to type in that little chat message. So the optic disc is this location where the optic nerve exits the back of the eye, and it contains no rods and no cones. That's why it's referred to as the blind spot. Okay, why is it the blind spot? There are no receptor cells. So think about it. If an image were projected right to this blind spot location, you don't see anything. Why don't you see anything? Because that image is being projected to a location on the retina that contains neither rods nor cones. You must have the image go to where rods and cones are in order for you to be able to process it. Okay? So that's why we have that blind spot. Now, what happens is your brain will complete patterns that fall onto that blind spot. So you're not aware of this blind spot in each eye as you walk around your world in an everyday basis, right? You don't see 2 pinholes in your visual system. Your brain sort of fills things in there for you. Finally, the optic nerve over here is basically the power cord. Okay, the optic nerve exits the back of the eye, and the optic nerve is going to project information onto other parts of the brain. Okay, the optic nerve, we'll see in just a minute here, is going to route visual information through the thalamus. Remember, we said the thalamus was the relay center for sensory information. Everything other than smell goes through the thalamus. I compared it to O'Hare Airport in Chicago, right? So we're going to send the message out the optic nerve. The optic nerve is about as thick as your pinky. Again, it's the power cord that comes out of the back of the eye, and right where it exits the back of the eye is where we've got this optic disc, or this little blind spot. Okay? All right, I can't keep track of how many different parts I've just noted for you there. It's probably 7 or 8 or 9. Any questions on the major structures of the human eye? Okay. I could give you a diagram and point at something and ask you to identify what it is. I could ask you, to be able to recognize the relationship between the iris and the pupil, or know something about what the pupil does in bright light versus a dimly illuminated situation, something along those lines. Let's go to Jasmine and Ashley for a couple of questions, and then we'll be ready to finish up the story on vision, and then turn our attention to the auditory system. Right, the fovea is cones and only cones. You got it. And Jasmine? Right, and the blind spot has neither rods nor cones. So if you throw an image right there, what happens? You don't see it. And in fact, your textbook, the physical book, has a nice demonstration of this where you can sort of line up your vision. You're looking at a black dot, and I think on the other side there's a picture of a car or a rabbit or something. And as you focus on the black dot, you can get the other image to project right onto your blind spot, and you won't be able to see it. Okay? The difference is, it's not really so much about a difference between pupil and iris, Thomas, is that they work together, right? The iris is the colored muscle that controls the size of that opening, right? The size of the pupil. So as the pupil constricts, that muscle is tightening up. As the pupil dilates and opens up, that muscle is becoming looser. Does that make sense? Good. All right. Any other additional questions about the hardware here for the visual system, parts of the human eye? Okay, let me bounce down and just show you where things go from here. This is more detail than you're going to be responsible for, but if we follow the pathways in the visual system through the brain here, we'll see the crossover, this optic chiasm, or the optic nerves, right? The power cords, as I described them, coming out of the back of the eyes, crossing over here, being routed through the visual area of the thalamus, left and right hemispheres, and then ultimately that visual information getting projected back to the visual cortex in the back of your head. We said that's the occipital lobe. That's where we do that later processing and sort of higher level processing, if you will. It's almost where we can think about perception taking place at the level of the cortex, that later richer processing of the visual info. All right? That's everything I've got on vision. I'm going to turn the page here, not literally, but turn the page and talk now about your auditory system. We're going to use this figure from your textbook to talk about the major parts of the auditory system. I don't have the figure number written down, and I don't have my textbook in front of me, but just like we had parts of the eye with that figure from your textbook, now we've got parts of your auditory system, and we're going to just follow the sequence here. As now in this case, auditory information is going to be processed, beginning with these things here sticking out on the side of your head. Okay, the technical term for those structures that hang on the side of your head that you put earrings in and so forth is those are called the pinna. And it wants to auto correct and say pinnacle when I type in pinna in the chat box, but that's the pinna. The job of the pinna is to funnel sound waves into and then down the auditory canal. I almost knocked my headphones off. Okay, the pinna will route sound waves from our environment into and then down the auditory canal. So you can see the sound waves here being gathered up by the pinna. So it's like a funnel, right? In fact, if you can't hear someone well, you're at something, some event where it's really noisy, you might even cup your hand like a funnel around your ear to expand the capture via that funnel, via the pinna. So it's pinna, and then this auditory canal, and the sound waves basically get routed down this road. So the auditory canal is basically a roadway from your outer ear to your middle ear, as you can see, outer, middle, and inner up above. Those sound waves will then start to bang on your eardrum. And just to get a better, cleaner look, we can look at this enlargement down the bottom of the middle and inner ear. Here's the eardrum. The other term for the eardrum is tympanic membrane. So let me try to type that in for you. The tympanic membrane is the eardrum. What does the eardrum do with those sound waves? Well, it's going to vibrate in response to the pressure from those sound waves banging against the eardrum. Okay? So, again, pinna funnels it in, it moves down the auditory canal, it bangs on the eardrum here. It bangs on the tympanic membrane. Excuse me. The eardrum's vibrations are further propagated through the auditory system here by rattling around these three little bones known as the hammer, the anvil, and the stirrup. I'm going to give you one term for all three of them. So that's good when you can get 3 for 1 here. Those are known as the ossicles. Okay, so the ossicles are going to vibrate in reaction to, well, the vibrations of the tympanic membrane, the eardrum. So it's just a sequence here, right? We're sending the message along. Pinna, auditory canal, tympanic membrane, and now these ossicles. The last of the ossicles right here, see where it says stirrup? That stirrup is going to press on the oval window of this structure here called the cochlea. If you look up above, the cochlea is sort of a snail-shaped, shell-shaped structure. Okay, it looks like a snail. It looks like a seashell. It looks like, if you're hungry, a cinnamon bun. It looks like Princess Leia's hair from the very first Star Wars movie back when I was about 6 years old. Whatever you want to use to remember the cochlea, you're going to want to remember the cochlea, because put a star next to cochlea. And because I'm telling you to put a star next to it, what do you think the cochlea contains? Tell them in the chat box. What do you think is contained in the cochlea that's going to be hugely important for hearing? The cochlea contains the receptor cells. Yes, the receptors for hearing are located in the cochlea. So down below here, they've partially uncoiled the cochlea just so you can see it better. It's actually curled up on itself like those various things I described before. What happens now is the stirrup, this final bone of the ossicles. By the way, these 3 ossicle bones are the smallest bones in your body. That stirrup presses on the oval window of the cochlea. Okay, then what happens? Well, you can see here the little arrows are following the movement of those vibrations. I've got another slide that I think actually shows that even better. So here is the stirrup pressing on the oval window, and you see the propagation of those sound waves through the cochlea. Okay, well, what the heck is this stuff over here? These are the receptor cells for hearing, and the receptor cells for hearing are called hair cells. Okay, hair cells line the basilar membrane here. If you look at this sort of fuchsia-colored segment here of the cochlea, you can see the hair cells lining that basilar membrane. Here's an electron microscope photograph of hair cells. They look like plants under the sea, maybe in your aquarium, growing up from the bottom and waving around as things in their environment press on them. Well, here's the deal. The hair cells are sitting on this basilar membrane, and the cochlea is actually filled with a substance called endolymph. You don't need to know what endolymph is, but endolymph is a substance that fills the cochlea, and endolymph is kind of like jello. Okay, so think about this. Hair cells are sitting on a basilar membrane which sits on top of a big pile of jello. And now I'm going to ask you a weird question. What happens if you press or push or bang on jello? What does the jello do when you push on it or press on it or slap it? It jiggles. It moves around, it jiggles and wiggles. That's exactly what happens here in the cochlea. If you imagine this brown basilar membrane here where these hair cells are sitting, getting jostled around from below because it's sitting on this jello, what's going to happen? The hair cells are going to be physically stimulated, and they are going to now transduce. They are going to convert. They're going to translate those sound wave vibrations into action potentials. And now, voila, the magic has been done, right? We've turned sound waves into action potentials. Action potentials are the language of the central nervous system we're often running, okay? What ultimately happens then is the sound waves are converted into action potentials, and the auditory nerve does for hearing what the optic nerve did for vision. Right? The auditory nerve right here is just the power cord coming out of now the inner ear, okay, the optic nerve right here, and it's going to ultimately send the message onto the auditory cortex in your temporal lobe. That's the final destination, if you will, for the auditory information. It's actually going to get routed through the thalamus first. Remember, thalamus sensory relay center. So let's follow the sequence, right? Pinna, auditory canal, eardrum, ossicles, cochlea, auditory nerve, temporal lobe, auditory cortex. That would be the full sequence here. So to help you pull this together, now that we've dug through thoroughly both vision and hearing, here's a little table to help you compare and understand the relationship between vision and hearing, right? I'm listing in the first column a sense. Second column, what are the receptor cells called? Third column, where are those receptor cells located? This is actually a table you could continue to build out as you read about the other senses in the textbook. So for vision, they're called rods and cones, and they're located in the retina. For hearing, they're called hair cells, and they're located in the cochlea. Okay, that's the story here for sort of pulling together vision and hearing. And the reason that I do this, guys, is that we know, in terms of students and memory and learning, that you learn better when you can relate concepts to each other. Okay, so you see me do this a lot in this course, where it's not just a bunch of separate individual little pieces of information, little nuggets, right? I like to pull those nuggets together and build something so that you can relate things to each other, right? Compare and contrast. Show the similarities and differences between things. Well, we've got similarities here because all of these sensory systems have receptor cells, so let's identify what they're called. All of those receptor cells are located somewhere, so let's talk about where they are, and then we're off and running, okay, to understand this stuff really well, really thoroughly. Let me stop for a second and go back to this slide that shows graphically the parts of the auditory system. Questions on any of the major structures that are part of the auditory system here, beginning with the pinna, running all the way through the auditory nerve, and then on up to the thalamus and the auditory cortex in our left and right temporal lobes. All right, Andrew has politely raised his hand, so the chat box is available for his question. What do the hammer and anvil do again? They're both sort of, Andrew, it's sending the message on. So the eardrum, bum, 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 bangs on the hammer, which presses on the anvil, which presses on the stereo, which then ultimately presses on the oval window. So it's sort of like a game of telephone, right? Message being sent from 1 to 2 to 3 to 4, straight on down the line. Okay, and remember, you can actually pull together hammer, anvil, and stereo, and just think about them collectively as the ossicles. If you understand ossicles, that's good enough. Make sense? Okay, good. Good question. Thank you. Other questions at this point on the major parts of the auditory system? And I got to say, you guys, with 60 plus, sometimes 70, 80 plus in here, you guys have been really, really good about utilizing the chat correctly and appropriately. I appreciate your efforts to do that. So Aaron has a question. While Aaron is asking that question, let me toss this little polling question out to you guys and see what you can do with that one. Rods and cones are to vision as blank are to audition. And Nia as well has a question. What parts of the ear are the first to fail after aging? That's a really good question. There's lots of variability, but certainly what you'd expect, Aaron, if something is going to go wrong and you're not going to be able to hear things, what do you think is the most important structure with the most important parts in it? Have you ever heard of cochlear implants? Exactly. That's where I'd go. If you made me bet on something, I'd say more often than not, it's probably going to be something related to the cochlea. My dad just had some of this. I forget which year it is. He lives back on the East Coast, in the Philadelphia area, and he now has a hearing aid, and I think it's related to aging and problems with the cochlea. Nia's asking about the eardrum. And what happens here is fake stirrups, nice. The eardrum is the tympanic membrane, and it is going to basically send on the vibrations that are coming in down the auditory canal. It's going to bang around, vibrate, and then cause those vibrations to make movements occur in the hammer, the anvil, and the stirrup. Those three parts, they are known as the ossicles. Okay? I'm sorry, I didn't pull up the responses to this one. Almost everybody went with hair cells, and that's really good. Okay, a couple of people are trying to change their answer now that there were 37 people answering. See, it doesn't matter. There are no points connected with this at all. Ava, you have a question. You can use the chat box for that. Without the pinna, it would be difficult because you wouldn't be able to funnel this information in to the auditory system, right? Without the pinna there, it would be difficult. So we go pinna, auditory canal, eardrum, ossicles, cochlea, auditory nerve. In fact, if you want an expanded version of that, I've got it right here on the slide. Okay? In fact, if we want to get fancy, we could add in a first one up here, which is pinna sends sound waves down the auditory canal. Okay, I didn't mean to make that all bold, but it just is. Okay, so pinna, auditory canal. Ossicles are going to magnify the sound, the vibrations of the tympanic membrane. Hair cells in the cochlea fire action potentials. I mean, if you had me pick one step that's most important, it's that, because without action potentials, the temporal lobe auditory cortex areas are not going to be able to do anything with what we were talking about there. Okay? Those sound waves coming in. The inferior colliculus. That's a brain structure we skipped in the section on biology because I was trying to be kind and merciful and not overwhelm you with 86 different brain parts. But it says here, this brain part, called the inferior colliculus, localizes sound in space. Anybody want to take a guess as to what that means? Take a shot at that. Localizes sound in space. What do you think? Tell me in the chat box. Separate sounds, finds the surround. You're getting closer. I want to localize the sound in my environment. What does that mean? Aha, I like it. Yeah, is it behind me? Is it in front of me? Is it to my left or to my right? Right? So locates the source of the sound. I think the best example to think of is when you're in a car and you hear a siren, right? Is the siren in front of me or behind me? Is it coming towards me or moving away from me? Might it be on my left or my right? And how do you figure that out? Tell me in the chat box. How do you figure out where something is? Do you ever notice what you do to help figure that out? What do you do with your head? You probably do this automatically. You don't even think of it. You look around, and if you can't see it visually, the turning your head actually helps you localize the sound. And the textbook writes about this a little bit as well in this chapter on sensation. So if you want to see more about that, that's interesting, I think. Thalamus then distributes frequency information. You'll read about this in the textbook. It turns out that the frequency of sound waves is what determines their pitch. I used to have separate slides on this, and I just felt like it was too much. Okay. Basically, the take-home message that you'll read about in the textbook is that higher frequency sounds yield higher pitch sounds. Okay. Higher frequency just means more sound waves pass a given location in a given amount of time. Okay. Higher frequency, higher pitch. Okay. So if you think of a singer who you think can really hit some really, really high notes, she is probably cranking out some really high frequency sound waves. If you think of somebody else with a really deep voice, right? That's just lower frequency sound waves. And your thalamus helps to distribute that information about the frequency of those sound waves, and we perceive that as pitch. And then finally down the bottom, we see the auditory cortex helping us to interpret sound. Okay, and I'm going to put down here in terms of that this is where we move on to perception. What do I mean by that? Well, as it says right here, interpreting sounds. What does that mean? That has to do with coming up with some sort of a mental image or an interpretation or an understanding of what that sound is, right? Sort of classifying it. Who is it? Is it my mom or my sister? Is it my uncle or my dad? Which one of my two brothers is it? Is that the sound of a kitten or a lion? Right? That's what the cortex does. That's this higher level stuff. And that really, I think, in a lot of ways represents this transition from the earlier, more biological sensation work that we've talked about all morning this morning, and then the later, more psychological perception work, which is what we're going to focus on when we get together on Thursday. Questions on any of this auditory stuff? Yeah, Mariah Carey is probably your best example of real high-pitched sounds. Right, high, very, very high frequency. Barry White, he's your low frequency sound guy, that deep sound. Okay, let's use our last 7 or 8 minutes here to talk about sort of where we're going to go next with sensation and perception as we move into specifically the discussion of perception. I want you to understand kind of where we're going to go here. So perception, as it says here, includes the processing, organization, and interpretation of various sensory signals. Look at that. Processing, organization, and interpretation. In fact, I think the most important word in that list is the interpretation of sensory signals. So again, think about the idea of a mental image, right? It says down below here, we end up with an internal representation of environmental stimuli. So I like to think about the idea of a mental image of something, right? So when we hear the sound of a siren and the of the fire truck, we almost get a picture in our mind of what that is. We don't scratch our head saying, I'm not sure what that is. That's a weird sound. No, based on our experience, and this is why perception is different, right? It's later. Based on past experiences with sounds, we know where to sort of drop that sound into our categorization scheme and understand it, right? We're going to talk on Thursday about lots of examples of perception. One of the neat things about perception is how what we've been thinking about influences how we interpret new things coming in. Okay? So if you've just been reading about topic A versus topic B, when I present you with stimulus C, you might interpret and process that new stimulus differently depending on whether you've been reading about or thinking about topic A or topic B. I'll give you some specific examples on Thursday for sure. Okay? So again, we're now transitioning to this later perceptual organization in such a way as to be able to make sense of new things coming in. Okay? Again, based on our experience, we're able to make sense of these things and, again, understand whether that sound is a threatening sound, the roar of a lion, rah, versus a little meow of a kitten. Okay? Being able to do that matters because it influences what we should do next. Here's the outline for this segment on perception. These are the 5 big ideas we're going to review together on Thursday. The first one is called bottom-up versus top-down processing, and we'll start with that, and ironically enough, that's probably the most difficult of the 5. When it comes around to testing time, that's the one that seems to cause the most headaches for the most people, being able to understand bottom-up versus top-down processing. We'll do some examples. We'll do a lot of examples of top-down processing, and probably by about 20 minutes into Thursday's class, you should clearly see the difference between those two. Okay? Concept number 2 on Thursday is going to be something called perceptual parsing. Really simple. I think actually the term is more difficult than what it's all about. So we'll talk about perceptual parsing. We'll talk about how we process things efficiently by following the Gestalt principles of perception. Gestalt is the German word for whole, W-H-O-L-E. So not like a hole in the ground, but whole, like meaning the entire thing. And we'll talk about how when we're looking, for example, with our visual system at a whole bunch of incoming stimuli, these Gestalt principles help us to efficiently process those incoming stimuli. It gives us some shortcuts, if you will, to understand all of that stuff that we're being bombarded with on a daily basis. We'll talk about some depth cues that we use in our perceptual systems, both monocular depth cues, which give rise to the perception of depth in a two-dimensional object. We look at a painting, we look at a drawing, and we see it in such a way as to think that some things are closer than others. We'll also talk about binocular depth cues as well. And the final little segment on Thursday, face-to-face, will be about perceptual constancies. Okay, so that's sort of the lay of the land as we make our way through this section on perception. We're going to be talking about these 5 big ideas. So that's your checklist for the content on Thursday. And if I look at my list of learning outcomes, it says number 2 for Chapter 6 here on sensation and perception, distinguish between bottom-up and top-down processing. There it is. Number 3 says explain Gestalt principles and concepts. I got that. Step 4 here, or learning outcome number 4, says describe binocular and monocular depth cues. So that's on the list as well. Okay? Whew. All right, that's a lot of talking, especially after I did it from 8 to 9:15 as well. So I'm going to stop for a second and see if we have any final questions on really anything from today. Anything about the test, anything about sensation in general, or specifically about vision, or specifically about hearing, if you have a question, you can just raise your hand. Nia, your hand is up. I think that might be from before. If it is, just click the hand again. It'll help you lower your hand. I don't want your virtual arm to get tired from that. That's okay, no problem. Any other final questions from today's session? I apologize for this quirkiness today. I don't know why PowerPoint wouldn't load directly into Collaborate as it typically does. Were you guys able to see everything okay, doing it in this weird kind of app share mode? I know it's a little bit smaller. I can't annotate it as much as I wanted to, but you know what? It's better than not being able to run the session at all. So I'm pretty good at doing this kind of MacGyver stuff to figure stuff out when we're struggling. So let me jump out here. I'm going to end the app share and finish up with that. It's going to toss me back into, I think, just a white screen there for you guys to look at. And I'll see if there are any final questions for today. You guys got anything else for today? Just toss that whiteboard there. Okay, so online office hours, I'll create that space for you to log into in just a couple of minutes here. Those will be from 11 to 12 today. We're done for today. We'll pick up on Thursday. Have a great rest of your Tuesday. Make sure you log out, because then that makes it so that I can speed up the processing and the making available of the archive, the recording from the session. All right? I'll see you guys on Thursday. Have a great day. Thanks.